My name is Justin G. Chitpin. I'm from the University of Ottawa, and my poster is titled Bayesian Annotations for Targeted Lipidomics or BATL, Accurate Identification of Lipid Species Quantified Using Liquid Chromatography coupled to tandem mass spectrometry via electrospray ionization in selected and multiple reaction monitoring modes. Targeted lipidomics based on LC-ESI MSMS excels in relative and absolute lipid quantitation using SRM and MRM modes. These methods monitor pairs of parent and product ions to detect lipid targets eluting within a retention time window. In SRM, a single transition is monitored per lipid target, while in MRM, more than one transition is monitored. Currently, no program exists capable of targeted lipid identification where single transitions are monitored per lipid species. SRM data is manually identified by analysts using parent and product ion mass overcharge and retention time, as shown in the extracted ion chromatograms below. On the left panel, a single peak is detected at this transition across both samples, and it is straightforward to see that these two peaks represent the same lipid identity A. More realistically, however, is the chromatogram depicted in the middle panel, where multiple lipid isobars and isomers are detected within a given transition and retention time window. Manually identifying lipids across samples is thus challenging, and any misidentifications may lead to biologically incorrect conclusions from any downstream statistical analysis. Furthermore, in the right panel, we observe that retention time alone cannot unambiguously identify the dashed peak in sample 2 without the aid of other features, such as peak intensity. These observations lead into my hypothesis that accurate targeted lipid identification is achievable by probabilistic modeling of lipid-specific peak features and statistical inference. To address this hypothesis, we developed the R package BATL, which uses a naive-Bayes model to estimate the posterior probability of simultaneously identifying all SRM peaks in a sample. In step 1 of the BATL workflow, exported SRM data is manually curated and concatenated into a single text file. In step 2, BATL imports this training set to construct the naive-Bayes model using the peak features specified by the user. Either a single model is constructed using these features, or BATL can exhaustively construct models from all combinations of features and export the best performing model. Here, we trained BATL using 8 features based on peak retention time, intensity, and shape. The naive-Bayes model to estimate the posterior probabilities of any joint assignment of lipid identities is broken down into 5 steps. First, the prior probability of observing each lipid identity is computed by maximum likelihood estimation. The feature likelihoods for each lipid identity are then computed using a Gaussian or log normal distribution. When computing the posteriors from the prior and likelihoods, transition-specific posterior cutoffs are also estimated for the unassigned lipid identity U. This process involves finding the smallest posterior probability of a correct peak assignment from cross-validation. Using the maximum weighted bipartite matching or MWBM decision rule, we formulate lipid identification as a joint posterior probability optimization problem, where lipid identities are assigned to the peaks that maximize this joint probability. In step 3, the trained BATL model can then be applied to label new SRM datasets. And furthermore, BATL is also capable of annotating lipid isotopes and sphingolipid-specific artifacts, such as dehydrations and deglycosylations. BATL was trained and evaluated using a sphingolipid and glycerophosphocholine SRM dataset consisting of 440 and 464 samples acquired from 4 time points, and each monitoring 129 and 165 transitions, respectively. The lipid identities in these datasets were structurally validated by SRM IDA EPI scans, and both datasets profiled several biological matrices across different neurological conditions and dietary treatments, and were equally stratified at random into training and holdout sets. Our first results showed that a combination of 8 SRM peak features in addition to retention time significantly improved lipid identification. Identification accuracy is defined as the proportion of correctly identified peaks over all detected peaks. These findings were validated by the holdout set, achieving approximately 95% identification accuracy or greater on either dataset. Furthermore, the unassignment rates using all 8 features were less than 1%, indicating well-calibrated posterior cutoffs. Given the strong classification accuracy, We next determined how few training samples were required for accurate lipid identification. As lipidomics data is specific to each lab's LCESIMS platform, every lab must manually generate their own training set to use BATL. Our second result showed that BATL indeed required very little training data for accurate lipid identification. Here, we retrained the BATL model using all 8 features with varying proportions of training data, resampled 10 times without replacement. Similar identification accuracies were observed in both datasets when using as few as 10% of the overall training set, with unassignment rates remaining below 5%, and this corresponded to 22 sphingolipid or 23 glycerylphosphocholine samples. In conclusion, we introduced the first targeted lipid identification algorithm for SRM and MRM data. We show that lipid-specific peak features additional retention time significantly improve lipid identification too. And furthermore, very few training samples are required to effectively classify peaks, enabling users to rapidly deploy BATL for fast lipid identification. BATL is available on our GitHub page and is trainable on any continuous feature supplied by the user. Finally, our approach generalizes to other targeted lipidomic data types, such as precursor ion scans, product ion scans, and neutral loss scans. I invite you to try BATL for yourselves, and thank you for your attention.